as Penny said, this is a talk that is a collaboration between the AT Society and Ataxia UK, and I'm going to be focusing on ataxia with oculomotor apraxia type 1 and ataxia with oculomotor apraxia type 2. So what we'll cover today, I will start off with an overview of AOA1 and AOA2. Then I will talk a little bit about the fundamental science and the biology that underlies the AOA1 and AOA2 conditions, and then move on to talking about the disease mechanisms involved in causing these two conditions. I'll then talk about some of the potential therapies that might arise from the research that's taking place in understanding the conditions further. And finally do a bit of a clinical research update. There have been a number of studies looking at ataxia symptom relief in other ataxias that could be of relevance here in AOA1 and AOA2 as well, and also some of the global initiatives that are ongoing that are very exciting. So starting off with clinical features and explaining a little bit about the inheritance of AOA1 and AOA2. So with AOA1, the age of onset is quite early on, so people start getting symptoms in childhood, generally between 2 and 10 years old. Whereas in AOA2, it's a little bit later, generally in the teens. However, studies have shown that there is quite a range, and the range reported is between 3 and 30 years of age. And AOA1 is caused by the APTX gene, and AOA2 is caused by the SETX gene. So looking at common symptoms, as many of you will be aware, the common symptoms is the coordination problems, the ataxia in the limbs and balance problems, slurred speech, oculomotor apraxia, which is a difficulty in eye movements, muscle wasting and numbness, known as peripheral neuropathy. Then there are jerky, sort of fidgety movements in the arms that in AOA1 they tend to settle as the condition progresses, whereas in AOA2 they tend to persist into adulthood. So talking a little bit about the inheritance, as you will be aware, AOA1 and AOA2 are genetic disorders which run in families, and they're inherited in an autosomal recessive way. So this diagram, you will see the father and the mother and the offspring, and the parents are normal carriers, so they do not have the condition themselves, but they carry the gene for the condition. So for each gene, you get 2 copies, and here you will see in red the mutated copy, and the same mutation is found here, and that is a change in the gene. So parents of people with AOA1 and AOA2 have 1 healthy copy and 1 mutated copy. When you have offspring, each child will have a probability of will have a copy from 1 parent and from the other parent. So if you happen to get this copy and this copy, you will get 2 copies of the mutation, and that means that the person has the condition. However, if you get this copy with this copy, or this copy with this copy, you get individuals that are carriers but do not have the condition themselves. And whereas if you get this one and this one, you get somebody who is not a carrier or does not have the condition. So essentially, with parents who are carriers, you have 1 in 4 chance of getting the condition. And that is called autosomal recessive inheritance. Now, talking a little bit about the biology. So how do we use DNA code to make protein? I'll talk a little bit about different mutations, and also importantly about DNA damage, because that is very important in AOA1 and AOA2. So how do we use a DNA code to make protein? So genes are made up of DNA, and here is an image of DNA. DNA contains the code to make the entire human genome, and it's made up of building blocks called base pairs, which are called adenine, thymine, guanine, and cytosine, and they're shortened to A, C, G, and T. And this is the form of the DNA double helix. So genes are made up of this DNA, and they are paired up. So an A will always pair up with a T, and a C will always pair up with a G. So from the DNA code, you then get a process called transcription, which means that you get to an mRNA. Now, the mRNA is extracted from the DNA. So as the DNA code is made up of these four components that I've talked about, the ACGT. The mRNA is similarly made up of components, and they're the same components, except instead of a T, you get a U, a uracil. So you will get this DNA code being transcribed into an mRNA code. And this mRNA code has a subcode to make protein. So the next stage is called translation, and here you get protein factories, so a number of different factors involved in reading this mRNA to assemble the protein. Now, the protein is also made up of a number of building blocks, and these are called amino acids. So for each 3 letters in the DNA code, each 3 letters code for 1 amino acid, and the order of the amino acids will determine what the protein is. Now, the important thing here is that the proteins are the workers, the things that do all the tasks and make sure that the cells function properly. So this is just a little bit of understanding of the biology of how you go from genes to proteins, or from the DNA code to proteins. So in a normal situation where you don't have a condition, the gene will produce the protein and you don't get the medical condition. Now, when there's a mutation in a particular gene, this results in either the protein not being made or being made incorrectly, and that will lead to a disease or a condition such as AOA1 and AOA2. So in AOA1, you get mutations in this APTX gene, as I explained earlier, and this APTX gene encodes a protein called aprataxin. Now, studies have shown that a number of different mutations have been found in different families worthwhile, and I think 16 different mutations have been identified causing AOA1. So they're all mutations in the same gene, but they're different mutations. In AOA2, as I said earlier, the mutations are in the SETX gene, and this gene encodes a protein called senataxin. And here you have even more. You have more than 100 different mutations in different places of the gene and in different individuals, which have been found in studies around the world. So different mutations still causing the same condition. Interestingly, there's a condition called ALS4, which is a type of motor neuron disease that is also caused by mutations in the same SETX gene. So it causes a different condition with different clinical features. Now, talking a little bit about these mutations. So they're all a change in the mutations all involve a change in the DNA sequence, and you can get different types of mutations. And here are a few examples. One type is called a missense mutation. So if you get a change in one of the DNA blocks that I was talking about, one of those letters, then you will get a change in one of the protein. And so by changing the amino acid, you get a different protein. Instead of having one change in the DNA block, another thing you can have is a deletion or an insertion. And the deletion or insertion can cause a frame shift. So for example, in terms of an insertion or a deletion, if you add something to the DNA code, then it will shift everything in the amino acids. So all the amino acids are then in the wrong order and you get an incorrect protein. That's the end result, essentially. So these are different types of mutations that you can get. Now, another important thing to note is that the inside of a cell is actually quite a dangerous place, and you get damage to DNA. And different types of damage can be caused, such as double strand damage. So if you remember, I explained that DNA comes in double strands. So you can get a break here in both strands, or you can get a single strand break just by breaking one of the strands. And there are naturally damaging agents within the cell, such as things called reactive oxygen species, and these are actually byproducts of our own metabolism, or external agents such as chemicals, ionizing radiation, or sunlight. So essentially within the cell, damage to the DNA occurs. However, what we do have is that we have protection against this DNA damage, which is critical for health. And there are DNA repair pathways in place to deal with the damage. And this is very important when we talk next about AOA1 and AOA2, this damage to this DNA within cells. So what are the biological processes that cause to AOA1 and AOA2? So one of the things that goes wrong is in this DNA repair. So I'll start off by talking the 3 slides on AOA1 and then 3 on AOA2. So the aprataxin protein, which is the one that is affected in AOA1, is known to be a component of this DNA single-strand break repair complex. So you get the break going on, and aprataxin is normally involved in repairing this. So studies have shown, for example, in AOA1, that depending on the type of mutation, the aprataxin is either depleted, reduced, or non-functional. So you either don't have it there or it's not working. And so it can't perform its normal function of repairing the DNA single strands. And so what you get in AOA1 is that you get an enhanced sensitivity to a range of DNA damaging agents that generate these single strand breaks. And other studies have also suggested that there is involvement of aprataxin in the double strand break repair. Now, the next system that has been studied is mitochondrial dysfunction in AOA1. Mitochondria are the part of the cell which is known as the powerhouse because they produce all the energy that's required for the cell. So what we find is that if there is a depletion of aprataxin, you get mitochondrial dysfunction, and you'll get an accumulation of DNA damage within the mitochondria. So what we know is that muscle and brain tissues are particularly vulnerable to problems with the mitochondria, to mitochondrial dysfunction, probably because there's a high requirement for energy by these tissues, by the brain and the muscle. And in fact, mitochondrial dysfunction of different types has been identified in a number of ataxias and other neurodegenerative conditions. So this is another pathway that has been studied in AOA1. Transcription is the system that I described earlier. So this is also affected in AOA1. So aprataxin is shown to have a role in transcription regulation. So if you remember, transcription is where you go from the DNA to the mRNA, this step here, before you then get to the protein. So aprataxin has been shown to interact with proteins directly involved in transcription. And it could ultimately lead to a deficiency in vital proteins and neuronal death. So these are 3 sort of pathways that have been studied in AOA1 that are showing a little bit about the function of aprataxin and what goes wrong in AOA1 cells. Moving on to AOA2, synataxin has many possible roles. So different mutations in the synataxin gene can cause either a loss or a non-functional synataxin protein, similarly to what we saw in AOA1. And studies have suggested a number of pathways that are disrupted within AOA2 cells. One is transcriptional regulation, which is similarly to what we've seen in AOA1. Another one is double-strand break repair response system. So like we saw previously, it's the system for repairing the DNA double-strand breaks is not working functioning normally. Other studies have shown a role of synataxin in another pathway, and that is the innate cell clearance system called autophagy, where, for example, you remove toxic proteins that are produced within the cell. So the cell has a number of things in place to deal with, say, toxic protein accumulation, and this is this innate cell clearance system. So a study showed that synataxin depletion, so when you did not have synataxin within cells, it inhibited the progression of this autophagy, of this clearance system, leading to an accumulation of toxic things within the cell. Another study also showed a role in controlling antiviral response. And finally, there is a role in R-loop regulation, which I'll talk about in the next slide. So R-loops are 3-stranded structures which form when this RNA hybridizes to a complementary DNA strand. So you get, during transcription, as I told you the process before, during particular points in transcription, you get the RNA, the DNA, and the other DNA, and it forms a kind of loop structure. And synataxin has a role in the regulation of these so-called R-loops. And what you find in AOA2 cells is that there is an accumulation of R loops that has a number of consequences, such as leading to defects in the transcription, in the termination of this transcription process, and also leading to mitochondrial DNA damage. So this is a study that's shown that R loops may have a role in AOA2, in causing AOA2 as well. So leading on to potential future therapies and gene therapies. So an important thing to understand, I think, is that all these studies on the disease mechanisms, on understanding the role of the proteins normally and how that is affected in AOA1 and AOA2, and the role of the proteins associated with each condition, is important, because then when you know more about it, you can identify potential therapeutic targets and then look at potential drugs that can be developed to target those problems within cells. So here are some examples. For example, in mitochondrial dysfunction could be targeted in AOA1. So a study in 2015 showed that a lack of aprataxin impairs mitochondrial function, as I described earlier, and this was treated with an anti-diabetic drug called rosiglitazone by affecting a particular cellular pathway. Now, this was not a human trial. This was a study within cells. But it shows, the potential for potential drugs looking at a particular pathway and trying to target it in that way. Drugs that influence autophagy, which is that cell clearance system, exists, or drug that targets the R loops in AOA2 may be potential things that could be looked at in the future as potential therapies to be developed. Now, another thing I wanted to mention in AOA1, which is very important, is that some studies have shown a deficiency in coenzyme Q10 in patient cells, in some patients. And coenzyme Q10, or CoQ10, is a naturally occurring antioxidant within cells, and it is important in the process of producing energy. And treatment with CoQ10 has been shown to help some patients. Some people respond positively. And in fact, in the Ataxia UK medical guidelines, this is one of the recommendations, as well as in the AT Society's leaflet on AOA1. So this is an important consequence in actual management of the condition. Now, looking at some future approaches, such as gene therapy. With AOA1 and AOA2 being genetic conditions, gene therapy is obviously an attractive potential therapy that could be developed. Now, gene therapy is a technique that modifies a person's genes to treat a condition. And it can be done by replacing the disease-causing gene with a healthy copy of the gene. And the gene could normally be delivered via an inactivated viral vector. Now, although there are not many gene therapy products developed, one really promising thing is that for another neurodegenerative condition called spinal muscular atrophy, we do actually have an approved gene therapy product called Zolgensma. And the active substance in this product it contains a functional copy of the gene, and when it's injected, it passes into the nerves, where it provides the correct gene to make enough of the protein and thereby restoring nerve function. So this is a really encouraging sort of story to show that in other neurodegenerative conditions, gene therapies have been shown to treat a condition. Now, another type of gene modification is antisense oligonucleotides, ASOs, and you may have also read about these. So antisense oligonucleotides are short single-stranded pieces of DNA. And here is a diagram. So in a normal situation, like we saw earlier, you saw the DNA being made into the mRNA, being made into the protein. Now with an ASO is this little piece of DNA that will target and will bind to the mRNA, and it will essentially be a different code. So it changes the code of the mRNA, and so you end up with a different protein. And the idea is that to make the change so that it becomes a normal protein in the case of AOA1 and AOA2. So this is another kind of approach that is being looked at in a number of conditions, and a number of ataxias have programs looking at the development of ASOs. And again, we have an approved product for this condition, spinal muscular atrophy. And there is also an AT ASO clinical trial ongoing, and I know I'm aware of other ASO clinical development programs ongoing, although there aren't any in humans yet. This AT one is actually in a person with AT. So these are kind of future approaches that might help in AOA1 and AOA2 treatment development. Now, I just wanted to talk a little bit about some of the developments in treating the symptoms of ataxia. So the next slides will focus not on the sort of more curative approaches such as the gene therapy, but looking at drugs that may alleviate the ataxia symptom. So the first one I wanted to talk to you about is a trial by a company called Intrabio. So they have a product which is N-acetyl-L-leucine, which is an orally administered modified amino acid. And there's a trial ongoing in a few neurodegenerative diseases. And recently the company announced some positive results from their trial in one of these conditions that is being used in the trial, Niemann-Pick type C. And Niemann-Pick type C is a neurodegenerative condition which has ataxia as one of the symptoms. So in the trial, they were able to show that their drug targeted the ataxia symptom, including showing an improvement in the SARA score as part of the trial. Some of you will be aware that the SARA is a way of measuring ataxia in the clinic. So it's a number of different tests are done, such as coordination tests and walking tests, and they're combined in an ataxia score. And that is often used in ataxia trials. And they're currently carrying out a trial in AT and hoping to also look at other neurodegenerative conditions. And the beauty of this trial is that they are hoping to actually find a drug that will help relieve ataxia in all ataxia conditions, not just the ones that they're trying in the trial. So that is their aim. So we shall see what happens with this trial. The other study I wanted to talk about is a different one, which is one involving a technique called cerebellar transcranial direct current stimulation. And Ataxia UK is actually funding a study with this technique in Italy. So recent studies by the group in Italy have reported that applying a low electrical current to the scalp may alleviate some symptoms in ataxia. And so they wanted to expand the trial, do it for a longer period of time, do it for a year, and in a range of different ataxias, and also with an increased number of treatments, so more stimulation over time. And the results are expected very soon. In fact, they have actually just come out. So we will be talking to our medical advisory panel and producing a lay summary of the results very shortly. And similar studies are also taking place around the world, showing that there seems to be an interest in this technique in a number of groups as a way of potentially providing some symptom relief. And the last trial I want to talk about is by a company called Biohaven, who are doing a phase 3 trial, which is supposed to be a last sort of final trial in the US, after which they would hope to get approval for the drug. And it's a 1-year trial in a range of spinocerebellar ataxia, and it's quite large with over 200 participants. The drug they have is called troriluzole, and the mode of action is that it's supposed to modulate glutamate, which is the most abundant excitatory neurotransmitter in the human body. So it's supposed to have some kind of effect on the nerve cells. And again, it may be useful in a range of ataxias, and we'll be waiting to see the results of this trial to see if they show benefit in the scars and then potentially in other ataxias as well. So these are the trials I wanted to talk about, and I just wanted to end on a new initiative called the Ataxia Global Initiative, which I think is really exciting, in particular for rarer conditions such as AOA1 and AOA2. So here's the logo. It's a worldwide platform for clinical research in the ataxias. So this was established by ataxia expert clinicians, and it started off by having 2 different networks. One was focusing on the spinocerebellar ataxias, which are the dominantly inherited ataxias, and one on the recessive ataxias called ARCA Global, where AOA1 and AOA2 would fit. However, recently there was a desire to combine it, so we now have a unique Ataxia Global Initiative, which is a network of worldwide ataxia specialists. Now, this initiative has an international steering committee, some office support from the office in Tübingen, and there's a website. I'm one of the patient group representatives on this steering committee. And the steering committee is truly international. We have people from really all over the world, which makes it very difficult to have meetings. But of course, it's really great to have representation from all over the world. And really, there are a number of different goals of this Ataxia Global Initiative. The idea is to try and agree on standardized data collection on ataxia patients to help with clinical trials. So there are a number of different working groups, for example, on brain imaging and on digital biomarkers and clinical measures such as the SARA scale, and to get everybody together, to get all these experts together in one room and really look at the data together and come up with some kind of consensus for everybody to use the same kind of measurements in trials. Another goal is facilitation of joint projects worldwide. So at the moment we have about 21 projects proposed, one of which should include AOA2 as well. So those should be on the website soon. So it's the idea is that, for example, if you wanted to do a project where you wanted to collect a number of samples from individuals to try and understand more about a particular protein, say the ataxin, then often you might need samples from all over the world, from more than a handful of patients that you can get one country. So the idea of collaborating worldwide is really, really useful for the rarer ataxias, and similarly for clinical trials. Another goal is education and training. And we were just talking, I was just at a meeting today, in fact, of the Ataxia Global Initiative, and we were talking about some potential training in some of these ataxia rating scales so that everybody can get training for more junior researchers, so you kind of get new people and new researchers in the field. We had an online conference in October as well. The last slide is a sort of another initiative that has some similarities to the Ataxia Global Initiative, but has also got some major differences. And this is a new Ataxia Consortium that's been created to accelerate the development of treatments for the ataxias. And it's a consortium in a not-for-profit organisation called the Critical Path Institute. And this is called Critical Path Therapies for Ataxias. Now this not-for-profit organisation has already worked in other conditions, other rare conditions. So it's great to now have a consortium specifically looking at ataxias. And it was launched very recently in collaboration with Ataxia Global Initiative, Ataxia UK, and the American Ataxia charity NAF. And the aim is to gain acceptance from the drug regulators, such as the FDA and the EMA, on the different outcome measures to use in trials. So it's often a struggle to know what to use to measure the ataxia or the condition in a trial. So what particular measurements are you supposed to use to show whether there is a benefit from the drug that you are trying in the trial? And to get the regulators early on and discussions early on is very important so that they could endorse what outcome measures to use in trials so that better trials can be run. And when it comes to approving the drugs, they are in a better position to approve them because they've already been engaged in earlier discussions and agree that these outcome measures used are the good ones to use. And this is funded by pharma companies. So showing the desire with pharma companies to fund this program means that they really feel it's a very useful thing to do for the ataxias to help in their drug trials in the future. So I think these last two initiatives, global initiatives, are really beneficial to the whole ataxia field. And as I said, in particular, for some of the rarer conditions, it's useful to piggyback on some of the work that is being done on some of the maybe more common ataxias.